Okay, so we left off, we were talking about the properties of water. And we talked about water, first off, sticking to stuff, sticking to itself, cohesion, sticking to other things, adhesion. Talked about water being good at moderating temperature, so the hydrogen bonds between water molecules cause it to heat up very slowly, but once you get it warmed up, causes it to cool down really slowly. So it moderates temperature nearby it. That leads to things like evaporative cooling also working, so sweating. We also talked about that water expands when it freezes. So hydrogen bonds solidify and it expands when it freezes. And then we talked about that water is really good at dissolving things, which is what we just went over in that last question. So ions, other polar substances will hydrogen bond with water and they'll stay suspended. So the final kind of property that we didn't quite get to in last class is that water participates in lots of chemical reactions, and it's usually a vital component to many chemical reactions. Lots of chemical reactions, it is simply the medium, so it's the substance that the reaction occurs in, because remember your cells are made up primarily of water, but it's also a participant in a lot of reactions. So a chemical reaction is when two or more molecules swap atoms somehow. They rearrange themselves and they make up new molecules, so they yield different molecules. Water is involved in most reaction and is often a reactant or a product. We're going to be spending entire chapters talking about two reactions, talking about photosynthesis, where water is a reactant in that process, and then we'll talk about cellular respiration, where water is a product of that reaction. Those are two of the major reactions that occur in living organisms. So photosynthesis, capturing sunlight energy, converting that energy into sugars, or using that energy to make sugars. And cellular respiration, where water is a product, is the process of taking molecules and extracting their energy to make it into a form of energy called ATP, to use that for cellular work. So water is a major participant in chemical reactions. The final little part about water, final thing that we're going to go into before we start talking about larger biomolecules, is the concept of pH. And really for now, I just want you to be familiar with what pH is a measure of, what it actually is, and know a little bit about the difference between when pH is high versus pH is low. What does that mean? So pH is simply a measure of the concentration of hydrogen ions relative to the concentration of hydroxide ions. So hydrogen ion is H+, hydroxide ion is OH-. What molecule do you get, do you think, if you have an equal concentration of both of them? Yeah, so you'd have two H's and one O. So H2O occurs, or is, basically the equal concentration of H+ and OH-. So H2O has what's called a neutral pH. pH of 7 is pure water. Has an equal concentration of H+ and OH-. If the concentration of H+, if these are not equal, and the concentration of H+ is high, and the concentration of OH- is low, we get something called an acid. Acids have a low pH. So low pH is high in H+ and low in OH-. Acids, things like lemon juice, like soda, like tomato juice, coffee even is acidic. So those are all acids. Acids have a high concentration of H+ ions. The opposite of that, if H+ is low and OH- is high, it's called a base, and that is high in pH. So pHs of 10, 11, 12, 13, like soap, ammonia, bleach, sodium hydroxide, those are all very high in OH-, so they have a high pH in their bases. And you'll probably notice here that a lot of cleansers, a lot of things that we use to clean are bases. A lot of cleansers are high in OH minus. And a lot of foods are high in H plus. A lot of foods have a fair amount of acid in them. So kind of a duality there. And water's right in the middle, right in a neutral pH. Where this is going to become important is that pH can have a large effect on chemical reactions. So you need certain pHs in order for certain reactions to occur. And actually one of the ways that your body's going to regulate which reactions occur where is by having different pHs. So you have a different pH in your stomach than you have in your intestines. So certain reactions that can occur in your stomach actually won't occur in your intestines because the pH is off. And a substance called an enzyme, which allows reactions to go forward, the enzyme that allows reactions to go forward in your stomach won't function in your intestines. And conversely, that's also true. So we're going to talk about the effects of pH as we go along. Even on Monday, we'll talk about proteins and pH, and we're going to learn more about kind of the effects of change of pH. But for now, I just want you to know the concept. So pH, concentration of different ions. A low pH is high in H plus and acidic. A high pH is high in OH minus. Okay? So that's that for now. Okay. This is kind of a summary question to see how well you understood the properties of water. So let's imagine a world where water was not as polar. So water does not have as strong a partial charge on it, in which case water does not form as strong of hydrogen bonds. Which of the following out of these 5 would result if the polarity of water was a little lower, or if a substance that was not as polar as water was what organisms lived in? So read through them and pick out the best answer. So let's go through these. So no one chose A. Heavier insects than water striders would be able to walk on the surface of ponds. Why is that? Why would that not be the case? Okay, so the whole reason why insects can walk on water is hydrogen bonds, or the cohesive nature of water is holding those molecules together and supporting the weight of the insect. If it was less polar, it would take less force to break them. So heavy insects would definitely fall through. Okay, B, increased cohesion would contribute to increased upward water transport in plants. So this is kind of along the same lines. If you had a less polar substance, are you going to have more cohesion? No, you're going to have the opposite. You're going to have less cohesion. So it's actually going to be harder to pull water up plants, and it's just like it would be harder to use a straw. So water would be less likely to stick together, so it's not going to be cohesive. C, the water temperature of ponds and pools would increase more slowly when in sunlight. Is that true? Okay. Do you know why? So you're right. Yeah. So heat, remember we talked about this, heat is molecules moving. And the reason why water takes a long time to heat up is that molecules of water are stuck together. So in order to move this one, I've got to move the one next to it. In order to move that, I've got to move the one next to it. So it takes there's a lot of force kind of keeping you from getting those molecules moving. If they didn't stick together as well, if they were less polar, then you could easily move one water molecule without having to move all the ones around it. You'd be able to heat it up a lot faster, but also cool down really fast as well. So water would heat and cool much more rapidly. It'd be easier to boil water. Okay. E, more salts would go into solution in blood and body fluid. So why do things dissolve in water? All right. Because salt is polar and it's attracting to the water molecules. So if you had a less polar medium than water, could you dissolve as much salt in it? No, you could still be able to dissolve things in it, but it wouldn't be able to support as much. It would take more molecules of that substance to keep the same amount of salt suspended. So kind of an analogy to this would be, let's say you're trying to pick up a car I don't know if this works that well, but and you had 10 super strong weightlifters, they could lift up that car. But if you had 10 elementary school kids, they probably can't pick up the same weight. They're not as strong. It's not as strong a factor. So you're going to need more of them. You're going to need 1,000, and they'll be able to pick it up. Right? So more water would be necessary to dissolve the same amount of salt. So we're left, if you process of elimination, you're left with D, which 50% of you chose, which good job, because D is correct. So sweating would be a less effective means of keeping cool. So if you don't understand the why of this, as you sweat, the molecules of sweat on your skin absorb energy, and they become more energized. And eventually they get so energized that they break free of the hydrogen bonds that are holding them to the other molecules of sweat and to your face, and they leave. And when they leave, they bring that energy away from your skin with them, and it cools down the average temperature on your face. The reason why sweating works so well is that they actually absorb a fair amount of energy before they leave. If it was less polar, then as soon as they start heating up, they'd leave. And they wouldn't have time to absorb a lot of energy before they get pulled off. So every molecule that evaporates off of your skin would pull off less water, or less heat, less temperature than sweating does. So you'd have to sweat a lot more. You'd lose a lot more water to sweat to cool your body by the same amount. So because water is polar, you don't have to sweat as much. That's basically the thing. The same amount of water pulls off more heat. Does that make sense? Okay. And that one's kind of a hard one to grasp, but you just think of water pulls off a lot of heat.
So
Yeah.
like if it were less polar, then like as soon as it escaped your pores, then it would be gone.
It would,
Wouldn't have as
yeah.
much water sitting on top of it.
Yes, you would definitely be drier, but you think if you look at each individual molecule that you've sweat out, would pull off less energy. So in order to cool yourself by the same amount, you would have to sweat more. So in that case, sweating is less effective. Yes, you wouldn't feel as sweaty, but you would have lost more water to get the same cooling.
So that's related kind of to see with like the state change and the energy absorption.
Yeah, exactly. And they're all related. But yeah, that would be here too. Yeah, it would take less energy to boil water. It would also take less energy to evaporate water off of your skin. So in which case the water takes less energy with it. Yeah. Good. Okay. So if you didn't get those, this is a good one to review and think about the different properties of water. So we have cohesion. Kind of the first two are cohesion. We have moderation of temperature. We have this, which is also cohesion as well as moderation of temperature. And then we have water being a good solvent there as well. Okay. So the next thing that we're going to go into is we're going to start talking about larger molecules. So we've been looking at atoms. We've been looking at really small molecules like water, carbon dioxide, ammonia, things like that. We're going to start building larger biomolecules, and that's what we're going to get into for the rest of today. But before we really start talking about the major biomolecules that make up living things, I'm going to talk a little bit more about carbon and about organic molecules. So organic molecules are what generate life's form and function. And organic molecules are primarily made up of 4 elements. And we kind of talked about this a few lectures ago when I first introduced the elements. These 4 elements, carbon, nitrogen, oxygen, and hydrogen, are what make up about 96% of organic matter. And if you look at the electron configuration for all of these, you can pretty quickly figure out how many covalent bonds each one can make, or what what's called the bond potential of these different molecules or different elements are. So hydrogen, which has 1 valence electron on a valence shell that can hold up to 2 electrons, can make 1 covalent bond. So hydrogen will only make 1 covalent bond, and that's the maximum it can do. oxygen, which has an atomic number of 8. It has, therefore, 6 valence electrons, so it's missing 2. It can make up to 2 covalent bonds. Nitrogen, which has 7 electrons, has 5 on its outer shell. It can make up to 3 covalent bonds. And carbon, which has 4 valence electrons, 6 total electrons, can make up to 4 covalent bonds. So, when thinking about these 4 major biomolecules, if I wanted to make a large molecule, these 4 elements, if I wanted to make a very large biomolecule, which one of these 4 would most likely be at the center of that biomolecule? Could hydrogen be at the center of a large biomolecule? So why not? All right, it can only make one bond. So because it can only make one bond, it can't be at the center of anything. It has to be on the edge. And that's what's going to happen. Hydrogen's always going to be on the outside of a biomolecule. It's always going to be in the exterior. Oxygen and nitrogen, they could potentially be at the center. If you had an oxygen molecule, all right, oxygen, that's not a very good marker. So if you had oxygen, you could have, 2 things come off of it, and you could make something that's a bunch of oxygens in line. If you had nitrogen, it's a little bit more. You can have 3 bonds come off of it. But if you really wanted to make a large, complex biomolecule, your best bet is carbon. Carbon can make 4 bonds. You could make these very large biomolecules that branch out in all 4 directions. So, and this is what we see. We see that most organic compounds are based in carbon. So carbon forms the base of organic compounds. This is why we say we are a carbon-based life form. And the reason why carbon can form the base is that it can form up to 4 covalent bonds. It will form double bonds, single bonds, and can make these very large, complex molecules. Here are 3 examples of carbon-based molecules. Here's methane, which is just a single carbon surrounded by hydrogen. Here's ethane, which is 2 carbons, so you can make lines of carbons. You can also make circles. So here's benzene. You can make a ring of carbons. Organic molecules consisting of only carbon and hydrogen, such as methane, benzene, and ethane here, are called hydrocarbons. So, hydrocarbons. And you can make very complex structures from just using carbon and hydrogen. So, carbon and hydrogen, hydrocarbons. Carbon and hydrogen, if you remember back from when we talked about electronegativity, their electronegativity is not all that different. So, within the covalent bond between carbon and hydrogen, there's pretty equal sharing of electrons. So what does that mean about this molecule? Is it polar or nonpolar? It's nonpolar, right? So if I was to put ethane, methane, or benzene into water, would it dissolve? No. No, it wouldn't. So it's nonpolar, it would separate. So what do you think I could do to change the structure of something like ethane to make it polar? Do you know of a bond that is highly polar? A covalent bond between what 2 atoms? All right, hydrogen and oxygen, hydrogen and nitrogen. If you added an oxygen molecule or atom, or you added a nitrogen atom, you could change this into a polar molecule. Adding something like oxygen and hydrogen, you're adding something called a functional group. So hydrocarbons are nonpolar and generally non-reactive. The addition of a functional group will change the properties of a hydrocarbon. So ethane, which was nonpolar, non-reactive, wouldn't dissolve in water. Ethanol here, which has an OH, the OH is polar. Ethanol can now dissolve in water. You now have a molecule that has a polar region, can interact with water, can perform function in a reaction now has a lot of new features to it. Okay, so functional group, that term refers to anything that you add on to a hydrocarbon that changes the function of the molecule. Okay. So your book goes into a little bit of detail about some of the different functional groups that exist out there. I'm going to show you a slide on 4 different functional groups. If you don't remember those specifically, that's okay. The only functional group that I really want you to remember and to really have a good understanding of is this one, is the OH functional group, because it's pretty important to know that adding an OH makes something polar, allows it to dissolve in water. Right? So that's really nothing else. That's the functional group I want you to know about. This functional group, OH, is called a hydroxyl group. So here are 4 functional groups. Hydroxyl group structure is OH. It adds polarity to a molecule. So OH. So here are 3 other ones. And we're actually going to see some of them today and Monday as we talk about different biomolecules. This one will become very important later on in the course. So carboxyl group, which is a C with a double bond to an O, also bound to an OH. Carboxyl groups, what they do is they make a substance acidic. So they are an acidic functional group. What happens when you put a carboxyl group into water is that the hydrogen dissociates and becomes H+, so it increases the concentration of H+ in the water that it's in. So carboxyl groups will make a substance acidic. Amino groups do the exact opposite. Amino groups, NH2, will actually absorb hydrogen. It'll actually pull this out of solution, so lowering the concentration of H+. So it's going to make something more basic. So carboxyl groups are acidic, amino groups are basic. But don't worry about memorizing that. And this third group, phosphate group, is going to be important later on when we talk about something called ATP, adenine triphosphate. Phosphate groups are really good at absorbing energy, of storing energy. So we're going to see how the structure of this, which is just a phosphate surrounded by a bunch of oxygens, some of which are negatively charged, is going to allow molecules to store energy. There are other functional groups out there. There are lots of other ones if you ever look them up. They all have different properties to them. They all change properties of a molecule in different ways. These are just some examples. Really, if you remember the hydroxyl group, that's enough for this course. Okay, so functional groups change the function of a hydrocarbon. So here's an example of how dramatic functional group change could be. So here are 2 molecules that are pretty much the same. They both consist of 5 carbon rings. These are all carbon, carbon, mostly hydrogen, carbon there. This one has a hydroxyl group at this point. This one has a ketone. It has just an O that's double bound to the carbon. And there's one additional CH3, one additional carbon and hydrogen group here. Other than that, they're exactly the same molecule. The one on the left is estradiol, which is related to estrogen. So it's a compound that gets converted into estrogen. The one on the right is testosterone. Is there a difference between the effects of estrogen and the effects of testosterone on a biological organism? Yeah, you probably know about that, right? So there's evidence of that in this room, that there's differences between estrogen and testosterone, right? So the small functional change, small change in functional groups can lead to large biological changes. So they will be something that's important to consider, especially if you dive more into biochemistry. You learn more about that. Okay. All right. So that's the basis of carbon-based molecules and the basis of functional groups. So now I think we're ready to start talking about larger biomolecules. Okay, so by putting together carbon and hydrogen and oxygen and nitrogen and putting them together in different ways, as well as some other elements that are important for life, you can make up 4 major different types of biological molecules. And these are what make up almost all of biological organisms. Carbohydrates, proteins, nucleic acids, and lipids. And we're going to spend the rest of today and all of Monday talking about these 4 classes of biomolecules, what makes them different from each other, what are some of their features, where do you find them, giving you a really good overview of these different biomolecules. The reason for this is that the next few chapters we're going to start to look at their function in an organism. So we're going to look at where these are found within a cell, where these are manufactured and broken down in a cell. Then we're going to look at how things like carbohydrates and even proteins and lipids and nucleic acids are all used in energy extraction, and energy conversions. And then we'll look at how nucleic acids are used for making proteins and how we're going to control a lot of the functions of a biological organism through those different things. So these are going to be the basis of pretty much the rest of the chapters that we're going to talk about in this course. To some extent, it's going to be one of these things. So knowing a little bit about their basic properties is a good way to start. 3 of these, the first 3, nucleic acids, proteins, and most carbohydrates, are composed of small units linked together into larger units. A small unit can also be called a monomer. So mono for one and mer for unit, so a single unit. If you link together a bunch of monomers together, you're going to get something called a polymer. You may have heard of the word polymer before. Polymers, poly for many, mer again for unit. So you can have a single unit and you can link it together into a larger structure that's called a polymer. So the first 3, carbohydrates, proteins, and nucleic acids, will do this. Lipids don't form polymers. They're the one biomolecule that doesn't form a polymer. They function in a slightly different way, and we'll talk about that on Monday. So how do we go from a monomer, a single unit, into a polymer, a larger unit? For all 3 of those, it's the same reaction, same general reaction. There's a little bit different between the chemistry of them, but for the most part it's the same reaction. It's called a dehydration reaction. So dehydration synthesis is the process of linking together monomers into polymers. And what does the word dehydration make you think of?
Losing water.
Losing water, yeah. And that's exactly what's happening in this. So it should be really easy to remember that dehydration synthesis pulls out a water molecule to create a bond. So here are 2 monomers. Monomers, although their chemical structures will be different based on which biomolecule we're talking about, they're all going to have an OH on one side of them and an H on the other side. They might have more things or more of these functional groups sticking out off of them. But an OH and an H. In order to link these 2 monomers together, this H and this OH will be removed. So you'll remove H2O and you leave behind a bond. So that's called dehydration synthesis. It joins monomers into polymers. The opposite reaction is called hydrolysis. It's going to break down polymers into monomers. And what molecule needs to be added back to break a bond? Water. Okay, yeah. So hydrolysis, hydro for water, lysis for breaking. So water molecule here is added in, and you get your OH and your H back, and you've broken that monomer down. So really important reactions to remember the names of, because dehydration is going to be what takes single sugars and links them together into carbohydrates. It's what takes amino acids, links them together into a protein. It's what takes nucleic acids and so it takes nucleotides and links them together into nucleic acids. So we're going to see that in all 3 of those classes of biomolecules. So dehydration builds, hydrolysis breaks. Dehydration is a removal of water to create a bond. Hydrolysis is the addition of water to a bond to break it apart. Okay, so now we're going to start going into the different classes of biomolecules, and we'll at least get through carbohydrates today. We might not get to the other 3 completely today. So carbohydrates, which many of you have heard about, they're very important molecules in our lives. They make up a lot of our diet, right? Are sugars and polymers of sugar. So sugar will be the single unit in a carbohydrate, and then you can link it together into a polymer of sugar. Each of these classes of biomolecules has its own name for its monomer and for its polymer form. In carbohydrates, the monomer form is a monosaccharide, so for single sugar, monosaccharide. And the polymer form is a polysaccharide. So you have monosaccharide, you have polysaccharide. There are other terms that you may hear and you may have seen in your book. If you have 2 units of sugar together, 2 monosaccharides together makes a disaccharide. And again, linking together 2 monosaccharides, like here on the bottom, glucose and fructose, you do dehydration synthesis, you remove this H2O, and you have now sucrose, which is a disaccharide. Polysaccharides are usually really, really long chains. There is another name that's used for an intermediate sized chain. It's called oligosaccharide. You may have heard that word before. So you have monosaccharide is 1, disaccharide is 2, oligosaccharide is many, but not as many as polysaccharide. Polysaccharide will be hundreds, very large chains. So here's some examples of monosaccharides. Here's ribose. This is important for RNA. Here's glucose, very important for our diets, and this is something we're going to talk a lot about throughout the semester. And fructose, which is a monosaccharide found in fruit. So what do you notice about ribose, glucose, and fructose in terms of what functional groups you can see? So you see, so what functional group out of the ones we've talked about? What functional groups do you see on these monosaccharides? Yep. Yeah, all of them have actually multiple hydroxyl groups. So you've got OH, 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 HO, something like that. So you've got OH groups. So do you think these will dissolve in water?
Yes.
Yeah, pretty quickly. They're very, very polar because they have lots of hydroxyl groups. So monosaccharides, very, very polar. As you link together monosaccharides into long, long chains, you get polysaccharides. Here's 3 examples of polysaccharides. Here's cellulose, which is a polysaccharide of glucose. Here's starch, which is also a polysaccharide of glucose, but a different form of glucose. And here's glycogen, which is an animal polysaccharide. So these are both plant polysaccharides. Cellulose, really important in building tissue for plants. Starch, really important for storing energy in plants. And glycogen, very important for storing energy in animals. So we have glycogen. If you eat excess sugars, excess carbohydrates, some of that might be stored as glycogen for you to use later on.
So are all monosaccharides polar?
Yeah. Yeah.
Like all of them?
Yep, all of them should be. The word carbohydrate, if you just break down that word, you have carbon and hydrate for water. They're all CH2O. They're all there's some multiple of that. So we have C6H12O6 is glucose. C5H10O5 is ribose. C6H12O6 is also fructose. It's just arranged slightly differently. So they have an equal amount of oxygen as carbon. So they're going to have a lot of hydroxyl groups. They're going to have a lot of polarity to them. Yep.
Would sucrose be a disaccharide?
Yes. Yes. Sucrose is a disaccharide. Okay. So your 2 monosaccharides together. Another disaccharide we'll talk about later on this semester is lactose. So lactose is glucose plus galactose. It's 2 monosaccharides linked together. And we'll eventually talk about why people that are lactose intolerant, often the reason why they are is that they have trouble doing the hydrolysis reaction to break lactose down into glucose and galactose. So, and when you can't break down a disaccharide, you can't digest it, so something else that's in your gut digests it, usually bacteria, and they often are very gassy when they do that, which is why people that are lactose intolerant, if they eat lactose, they get gassy. It's because bacteria are digesting their food, not them. We'll talk about that later on in the semester. Okay, so some of the major roles of carbohydrates. So one thing, just kind of mentioned it, glucose is one of the most common monosaccharides, and it's something that we're going to use as our kind of example of a monosaccharide throughout the semester. Carbohydrates are a major fuel source, and we're going to talk about glucose as a fuel source. There are other monosaccharides that are fuel sources. in animals and plants, but we're going to mostly talk about glucose so we can kind of focus on one. In addition to being a fuel source, they're also raw material for building molecules in both plants and animals. So grasses, most of what you're seeing is cellulose, which is a plant polysaccharide. In insects, there's the exoskeleton of lots of insects is made up of chitin, which is also a polysaccharide. It's an animal polysaccharide. So there are carbohydrates that we use for food. There are carbohydrates that are used for structure. Kind of an interesting aside here is cellulose, which is a polysaccharide, which is of glucose, versus starch, which is also a polysaccharide of glucose. There are only slightly different structures to them. It's just the way that it comes together. One's called alpha glucose, one's called beta glucose. We can't digest cellulose, but we can digest starch. And it has something to do with the enzymes that are present in our bodies. Animals that do digest cellulose, they usually have a relationship with the bacteria that can digest cellulose. So cows, the reason why they can eat grass is that in their stomach, and they have multiple chambers to their stomach, they have a bacteria that's growing that will feed on the cellulose and break it down into monosaccharides, break it down into glucose. That's why cows will be chewing for long periods of time. They swallow it, they then regurgitate, and they chew some more. They swallow it, they regurgitate. And what they're doing is they're actually aiding that bacteria by breaking it down physically, and then aiding that bacteria to digest the polysaccharide down into monosaccharides. And then they'll swallow it into another chamber of their stomach where they will digest the monosaccharides as well as some of the bacteria as well. And they'll live off of that. So they're not actually doing the primary digestion of their food. What they're doing is they're sort of farming bacteria in their gut, which is crazy, but that's how they do that. And lots of organisms, lots of animals that eat grass, that's what they do. Okay. So here is another question. Okay. So the molecular formula for glucose is C6H12O6. What do you think the molecular formula for a molecule made by linking 3 glucose molecules together would be? So linking them together by dehydration synthesis. A lot of you forgot something, but a lot of you thought this was a lot easier than it actually was. But you should have thought there's got to be a trick to it, right? So if you just multiply it all by 3, you get C18H36, H36, O18, right? But what does a dehydration reaction do? All right, removes water. So here are 3 glucose molecules. They have other parts to them, but I didn't draw them all. And in order to link these 2 together, I need to remove H2O. In order to link these 2 together, I need to remove H2O. So in order to link 3 molecules of glucose together, I'm going to be removing 2 molecules of H2O. So 2 oxygens, 4 hydrogens. So I'll have C18, but I'm going to have H32O16, right? So that would be B. Right? So some of you got it. It's about half and half. I don't know, some people guessed these, which I don't know how you came up with those numbers, because I just randomly put numbers together, but somehow you thought those were right. But at least most of you were on the right track. You did your math properly. But don't forget about dehydration. So dehydration reaction removes water. That's the whole point of this. It's not the kind of question that I'd ask you generally. All right, I'm just going to briefly, we really don't have any time for this. What we're going to do, we're going to start on proteins on Monday. We're going to do proteins. We'll talk about lipids, and then we'll talk about nucleic acids, so the last 3 of biomolecules. So we'll save that to Monday, and that'll be the last part of Chapter 2.